In the interest of time, we do need to move on to Cole. Cole is our next speaker. Zmurchok. Yeah, great. From Vanderbilt University. So he's going to be talking about mechanosensing can enhance adaptation to maintain polarity of migrating cells. Fantastic, full screen, it looks great. Oh, but you're muted.
Excellent. Okay, yeah, thank you. So I'm going to switch gears from DNA back to continuum mechanics in the scale of cells. I'm Cole Zmurchok. Right now I'm a postdoc at Vanderbilt University in Nashville, Tennessee. Just like to thank the organizers for hosting the conference. It's been great so far. I just want to first advertise that this has been a joint work that I've done with my postdoc mentor here, Bill Holmes, and two people in the biomedical engineering department in the University of Melbourne, Jared Glett and Vijay Rajagopal. And it's published in Biophysical Journal, so the paper is out if you want to take a look. The background for the sort of problem I'm thinking about today is cell movement and how cells adapt to their environments. And so, as Julia already mentioned, that the movement and the shape of the cell is largely controlled by the cytoskeleton. So here's an image of one of these famous images, actually, of a migrating cell that has a large sort of branched actin structure in the front, the nucleus, and the cell body. And so the cell is able to migrate forward by basically pushing itself forward, pulling its rear towards the front, and maybe sticking to the substrate. Now, the cytoskeleton itself is a dynamic structure that can be sort of rearranged. Think of it as like a scaffolding that can move. And there's a whole bunch of proteins and other sort of receptors, and a whole bunch of different things involved in regulating this actin cytoskeleton. So here, what I'm just trying to emphasize is that there's this sort of a circuitry to the cell signaling that regulates the cytoskeleton. And at the core of that, there's a series of central regulators, GTPases, row family GTPases, that act as little molecular switches. So they switch between an active and inactive form. When they're active, they're actually bound to the membrane, and then their downstream effectors take place. So for example, when this protein rack is on, all of the good stuff gets activated downstream of it, and the cell starts to polymerize actin and actually expand. And when it's turned off, well, it's just turned off. Now, the data that motivated our modeling work is from the lab of Ryan Weiner, who's University of California somewhere. And what you're going to see here is Merck's image of fluorescently reported bioreporter for rack activity. And the cell that's just doing its thing sitting on a dish gets dumped with a very high level of chemoattractant. So the rack activity pathway can be activated by external stimuli, such as chemoattractants. And you can see the cell that's initially polarized gets some stimulus. Rack activity fires up and becomes highly active all throughout the cell. As time progresses on, you can start to see that there seem to be two polarity sites that form, and they sort of compete. Eventually, the cell resolves the fact that it's being sort of stretched and polarizing in two different directions and ends up taking a single polarized front. And that's indicated by that little white arrow in the last panel. And so what I mean by polarized here is that we have an area of high rack activity at one side and an area of low rack activity at the other. And so this cell you can think of would be building actin polymerase in the front, moving forward, and not doing so in the rear. Now, the other interesting piece of data that motivated our work is that not just signaling is one part of the story, but it's not all. Other important things like mechanical feedback are incredibly important. And I think Julia's talk earlier was a great example of how cells respond to mechanics in a literal way. And so there's another little piece of evidence showing that this rack signal can actually respond to an increase in tension on the cell. And so what you can see is a cell outlined in yellow. rack activity again is reported as that bright white spot. And the poor cell is being basically sucked on with a little pipette. So they're able to increase the effective tension on the cell membrane. And as they turn the tension on, the vacuum cleaner turns on, that rack signal disappears. Okay, and so this is starting to show us that this influence from mechanics can actually modulate these biochemical signaling pathways. So one of our hypotheses for how this cell that gets really highly stimulated can actually adapt to that sort of oversaturating stimuli, that oversaturating input, is that this mechanosensing process by which that the cell is able to sense that it's being stretched can then modulate the underlying signaling network and allow the cell to adopt a polarized signaling pattern. Now, what we did to probe that is we encapsulated the big rack signaling network that I showed you earlier, sort of with its bare bones minimal circuitry. That is, that rack switches between active and inactive forms, membrane bound and freely diffusing. So there's a difference in the diffusion coefficients between the two things, as well as some nonlinear positive feedback from rack to itself. And there's a whole bunch of evidence that shows that rack signals to other players, other protein species, that enhance its own activation. So together, we formulated a set of reaction diffusion partial differential equations to model rack activity in a spatial domain. Let me just point out a couple things. There is a inactive rack, R sub I, becomes activated with this rate in parentheses. There's a basal activation rate B, this positive feedback term here, and a deactivation rate given by delta R. Okay, and so the basic question I wanted to ask is, with this model, this model's been well studied by many people in the sort of cell polarity field. How can we understand the dynamics of rack in this model in the context of the mechanical stimuli and the chemoattractant stimuli? So what I did first was I mapped out the spatial patterns that you can get using a tool called Local Perturbation Analysis. And the short story is that there's 4 different behaviors, or three that can occur. A non-polar regime, where for example, for large basal activation rate, but small deactivation rate, you just end up with a cell that has high rack activity all across the cell. And that would correspond to a cell that is kind of expanded and growing. On the other hand, inside the blue bubble, what you can get are so-called polarized patterns, where it's possible, upon stimulating a cell with a stimulus, to attain, for example, a rack activity profile that spatially varies. That's high in one area of the cell and low in the other. Okay, and the reason I focus on these 2 parameters is because, remember, the basal activation rate, chemoattractant is like turning the basal activation rate on. So you can imagine if you are a cell and you're sitting somewhere within this polarized bubble and you get stimulated, all of a sudden your basal activation rate might increase, causing you to enter this high rack activity state. Okay, and we know that tension can turn rack signaling off. In other words, one possible feedback is that mechanical tension on the cell could increase the deactivation rate. And so the cell is able to resolve this overstimulation by increasing its deactivation rate so that it ends back up inside the polarizable regime somewhere. Okay, and so this is the basic idea that I'm going to exploit in sort of my what's to follow. Now, let me just say one quick thing about this local perturbation analysis for those who are interested in understanding how patterns form in partial differential equation systems. It's not quite the standard well-mixed analysis or linear stability analysis that you might do to sort of find Turing patterns in one of these systems. Instead, the perturbation we apply is a small localized perturbation against the constant activity backgrounds at steady state. And by exploiting the fact that there's a difference in the diffusivities between the slowly diffusing active forms and quickly diffusing inactive forms, we're able to derive a system of ordinary differential equations, which are more usually studied using sort of standard bifurcation analysis tools. So this lets us build this map of parameter space and understand whether or not the cells will respond to stimuli or not. So what I did first was I basically just did that experiment I drew it on the parameter space map by changing the parameters in the PDE system over time. I first applied a chemoattractant by increasing the parameter B. This causes rack activity to spike. That's a bright yellow shown where it says max B in the chymograph on the right. And then over time, I allowed tension to slowly build up, pushing the system's signaling parameters back into the polarizable bubble. And as that happened, eventually what happened is polarization results. And voila. So this is one way that this mechanosensitive adaptation could work to resolve these sort of overstimulating events. And really, that's facilitated by the shape of this bubble. So it's sort of a generic model for how the signaling system is set up that moving over and up can help resolve these stimuli. So to push this a little bit further, we did a series of things. First, we built a simple 1D model of the signaling and mechanics, where rack can actually cause the cell to expand, right? Because rack causes actin to start polymerizing, it will cause the cell to expand. As the cell expands, it will increase its tension, corresponding to sort of its preferred rest length. That increase in tension feeds back into the deactivation rate, increasing it, which should resolve the rack sort of overstimulation. And so this is built in a simple sort of Kelvin-Voigt element where I have a spring and dashpot together. I numerically solve the partial differential equations in a moving domain that's freely allowed to change size. And when I do this experiment, I can look at what happens with feedback from tension and without feedback from tension. So first, without feedback from tension, if you stimulate the cell with this high level of chemoattractant by increasing this parameter B, the cell just expands a little bit and doesn't do anything fun. However, as soon as you add this feedback from tension and you allow the cell to sense that it's getting bigger, the cell changes its underlying signaling kinetics to push them back into the polarizable bubble, and eventually it polarizes and begins to migrate on its way. So that's what you can see in the middle chymograph. Now, one other interesting observation, kind of a prediction of this model, is that if you change the parameters, you're able to attain a sort of polarization relaxation oscillation, where the cell sort of alternates between a period of polarized and directed movement, and then sort of like a little period where it just relaxes. And this is very much similar to other kinds of motility, instead of polarized directed motion, but sort of like a stepping motility, which is observed in other cell types. So here's the sort of fun part, I think. The 1D model doesn't really capture the cell mechanics that well, nor does it capture the influences of geometry or shape. And so we thought that we would model the cell with a 2D continuum mechanics model. Sort of, you can think of this as maybe the bottom part of the cell that's adhering to the substrate. And we keep track of the displacements, the stresses and the strains. We solve the reaction diffusion PDE inside this domain, and again, allow rack activity to impose protrusive forces in the normal direction to the cell. Now, the Jared and Vijay, our biomedical engineering collaborators, are much better at this continuum mechanics than I am. And so I'm not going to go through the equations, but basically, it's very straightforward linear viscoelasticity for the model. So the first equation is like a F equals ma. The stresses balance with the displacement. The elastic and viscoelastic stresses separate easily. We have linear models for how those depend. Small strain approximation to compute the strain on the body from the displacements. And yeah, so we put that together with some estimated parameters from literature, and lo and behold, we can do the same experiment. So I can take my cell, and without feedback from tension, rack activity fires up, it gets a little bit bigger, nothing much happens. However, when you allow the cell to sense this overstimulation, the cell quickly polarizes, changes shape, and then undergoes directed migration. I just want to emphasize that although this x-axis is like 0, 1, 2, 3, the scale and the migration speed is correct for cells that are migrating. And so we have a pretty good estimate of the Young's modulus and the Poisson ratio and all these other parameters that govern the motion of these cells. So lastly, we thought to, once we resolved basically how this mechanosensing could work, one of the things that we wanted to say was that we think that this could actually work to help cells adapt to variable environments to facilitate their migration. And so we put two cells side by side. They don't interact. The one at the top here does not have feedback from tension. The one at the bottom does. And we put them in a chemoattractant gradient. So you can think of this parameter B as being gradiated over this domain. And what we would expect is both cells should polarize because this B parameter is the gradient. However, the one with feedback from tension should be able to resolve the sort of higher and higher and higher and higher levels of feedback that it gets to maintain its polarity. Okay, and that's exactly what we see. Both cells polarize and move. The cell at the bottom is able to migrate much further before this adaptive system is sort of swamped and its migration eventually stalls out. Okay, so just to summarize, we coupled a reaction fusion model for rack dynamics and a minimal model for cell mechanics, both in 1D and 2D. We understood how this signaling dynamics basically shows us how this adaptive response could work, and that these 1D and 2D simulations confirm that these cells can actually adapt to these high signaling levels. I just want to emphasize that we're not saying that this is definitely what happens. There's many other biochemical mechanisms for adaptation, but here this is another pathway that can be explored. So I just want to close by thanking my collaborators, as well as a friend of mine, Andreas, from the University of British Columbia, who's working on a very similar thing at the same time, as well as funders. Thank you.
Great, thanks so much, Cole. Any questions from the audience?
Sorry, maybe I missed that. So, would you be able now to test this model?
Yes. So there's two things. One is a prediction basically between essentially cell area, migration speed, and sort of the rack activity. Okay. That's kind of the fundamental prediction that comes out of the model, which you could, I think, if you just go and observe cells and measure the area and measure the activity of rack through one of these biosensors, you could see to what extent that lines up with the sort of time courses that we predicted. Now, I don't know how to do the experiment which would really test this. So the particular feedback from whatever tension is doing to the rack pathway is not well characterized. There's many different sort of players in the actin cytoskeleton and things like when the membrane unfolds, perhaps
are they, in example, inhibitors of this protein, or in example, can you knock it out?
Yeah, I mean, this is the thing, right? There's this big signaling network has lots of feedback loops, and when you it's not quite as simple as perturbing one thing and seeing what happens. But yeah, you can play with the cytoskeleton, for example. You can reduce the actin, how it polymerizes, and this may have some feedback on the rack signaling. But again, I don't know how to do that. We thought a lot about this particular, testing this mechanism for the tension. And I think as soon as the community of experimentalists gets a better handle on specific sort of molecules and pathways from stretch or tension on the membrane of the cortex to the cytoskeleton or the GTPases, then we'll be able to sort of test these things with more careful things. But yeah, I don't think the data is there yet to. I don't think it's there yet.
Thank you.
Great, thanks. I think it's about time to move on to our last speaker. So thanks very much, Cole. It was a great talk.